Doctor, we are live now, doctor.
Hello and welcome all of you for today's live webinar by Glaucoma Society of India. Today we have our international guest lecture, none other than by our very good friend, Professor Kahuk Malik. All of us know about his mixed procedure. We all do that in daily procedure here, the KDB blade, Kahuk's dual blade. Thank you so much, Dr. Kahuk Malik, for accepting our invitation. Without any hesitation, he just accepted our invitation. Thank you so much for your acceptance. Without much delay, we would like to hear from you on two important topics. One is on the advances in glaucoma therapeutics by his spyglass, I hope so, and on his blade, the KDB blade, we all do it here. So these two topics is going to be covered by Professor in 45 minutes time, 40 to 45 minutes time. And then we have 15 minutes of time for the discussion. May I request Dr. Siddharth Dixit to introduce today's speaker, Professor Kahuk Malik. Over to you, Siddharth.
Thank you, Dr. Satyen. It's an awesome gathering here, and it's a pleasure and privilege to introduce Dr. Kahuk. And who doesn't know him? Does he need an introduction? Well, for formality's sake, let me do it. He is the Slater Family Endowed Chair in Ophthalmology, Professor and Vice Chair for Translation Research, Chief of Glaucoma Services at Department of Ophthalmology, University of Colorado, and co-director of Glaucoma Fellowship, UC Health. And that's not it all. He also has more than 500 papers, chapters, and published manuscripts. And if you don't know him already, you will know him as the editor of Chandler and Grant's, my favorite glaucoma textbook. And believe me, you can read it like you read a poetry. And he pays significant amount of attention to educating everyone. And he has a website which you must check. And he's been a very renowned innovator for a lot of products, which includes a device for performing rhexis in corneal scars and other conditions, the dual blade, which we all use and we all know as the Kahuk dual blade, and a very interesting device, Streamline, and a special device that he is going to talk about today. And he has been honored by everyone. He's not only known amongst us, he's honored by everyone, including the American Glaucoma Society, when he started with, and later on as an innovator, as by the American Academy of Ophthalmology with the Senior Achievement Award. And what is very, very special for a clinician scientist, the Ludwig von Salman Clinician Scientist Award by Arvo long time back. So he is someone who has achievements on his cap, like every one of us has here. And he's a clinician scientist par excellence, and it's a pleasure to have you. All over to you, Dr. Kahuk.
Thank you so much, Siddharth and Satyan, for the invitation. I'm excited to share some of the technologies that we're working on. I'm going to go ahead and share my talk, and hopefully you'll have access to it here. Looks like you have a good view, so I'll go ahead and get started. When I got the initial invitation to speak today, we were talking about different topics, and there are two very different topics to discuss. One is the MIG space, and particularly KDB, that we've worked on extensively here at the University of Colorado. And then the other stream of discussion here will be around new horizons in glaucoma therapy. Basically, what's new, what's coming, what might be the next frontier of glaucoma therapeutics, similar to how the MIG space, when it first started becoming more popular. So that's really the effort that we'll be discussing today. Let's see if I can get in here. Okay, so these are my disclosures. And let's just start off with a quick outline. The first part of the talk will be around adherence to therapy and how some of the surgical devices can intervene when patients are not controlled on their therapeutics. And then we'll switch gears and we'll talk about some of the pros and cons of the different drug delivery approaches that have been explored to date, both on the market and those that didn't make it to market. And then the last part is me focusing on two technologies that we developed here at the University of Colorado. We all know about non-adherence. So if we're seeing patients, we know that the majority of our patients do not take their drops as we prescribe. It could be for several reasons like cost, physical limitations, just patient having a hard time putting the drop on the eye, the hyperemia, stinging, visual disturbances that can happen from topical therapeutics are all listed under adverse events and can make patients who don't notice any visual field issues less likely to take their drops. There's also inconvenience, forgetfulness, and lack of understanding. Our patients don't always understand why they're taking their drops. Adherence to therapy, we know from multiple studies, can be as bad as 30 to 80%. And I can tell you in our studies, most of them are 50 to 80% as far as the poor adherence to the therapy. And of course, if you're not treating the chronic disease, then you're likely to get worse. The treatment algorithm when I was in training looked very much like this. So this is year 19 out of fellowship. I've been out for a little while, as you can tell from some of the gray hairs. And this was exactly what we were taught to do. Mild disease, initial diagnosis, start drops. After the drops don't work, then you go to ALT or SLT, and then it progresses to filtration surgery, trabeculectomy, then of course a drainage device and CPC. This was all disrupted when MIGS came out. And MIGS sort of came in the middle of this and disrupted the continuum, where mild to severe disease can now be addressed by different combinations or freestanding MIGS devices, whether it's implantables like the HydroStent on the upper left-hand side, or some of the other devices that were mentioned in the introduction. We also have different ways of doing filtration surgery like Zen and PreserFlow. Now, when we're treating the patient, we're not just looking at, is this the first initial visit? Should I start on drops? We're looking at the entire patient and trying to tailor our therapeutics for the specific patients based on age, intraocular pressure, visual field status. Is it advanced or is it just mild disease? What type of glaucoma? We also know that some of these devices work better in specific subtypes of glaucoma. And it's really fun to be a glaucoma specialist today. We're seeing a lot more in the US, we're seeing a lot more of the residents getting into fellowship training in glaucoma because now they're seeing more than just trabeculectomy and glaucoma drainage devices. So let's talk a little bit about excisional goniotomy, which is one of those devices living in the MIG space. This is a device that we developed here at the University of Colorado, and I'd like to go through the history, just kind of walk you through how we developed this device, and then some of the clinical data and how we're using it today. And then from that point, I'll go into some of the new devices that we're working on here at the university. So the history of goniotomy is quite extensive. When you start looking at our literature, it might appear that we've only been talking about goniotomy since the early 2000s with the introduction of some of these devices like trabectome that came in 2004. But in truth, you can go back to the 1800s and see mentions of different forms of incision to the trabecular meshwork, culminating in Barkhan's publications in the 1930s and 1940s. A lot of these devices were studied in both pediatric and adult patients, and the adult patient data was not as promising. So they fell to the wayside, and it wasn't until maybe the past 15 years or so, 20 years or so, where goniotomy started seeing its way back into adult patient care. The genesis of the idea for KDB was, like a lot of things in medicine, not actually our focus. So we did not enter into this. I can tell you, I wasn't in surgery thinking I'd like to make a dual blade. That's not how it started. The way it started was some of the imaging work that we were doing, two-photon microscopy, trying to harvest strips of trabecular meshwork in order to do precise imaging. And I found very early on that it was extremely difficult to get a strip of trabecular meshwork, which behaves very much like wet tissue paper. So I thought, okay, maybe I can design a device that would help our lab group in harvesting trabecular meshwork. And I started working on different iterations looking at making dual incisions that were parallel to create a strip automatically. We were able to do this, and you can see some of the steps that went through. When we got to that final design and we were working on it for harvesting trabecular meshwork, the thought immediately went to taking this into the operating room as soon as we can. It was quite effective at removing strips of trabecular meshwork, leaving very little of the residual leaflets behind. That we got very excited about the utility. But the battle was in trying to design exactly the dimensions that were needed. And I can tell you, if you look at the design of dual blade, one of the hardest things to achieve was the degree of the ramp. We wanted to make sure that we could reach in under the canal, in the canal, and lift up the trabecular meshwork so that before getting cut, the trabecular meshwork was above the plane of where it normally sits. Essentially, what that did is it put the trabecular meshwork on stretch, and it allowed for very clean dual incisions to remove the strip in a very routine way. So it seems now, after having designed it intuitive with the ramp and the dual blades, but I can tell you it took a lot of multiple iterations to get to the right dimensions and then to get to the right spacing of those blades so that it could accommodate various forms of glaucoma and various anatomies. You can see some of the features here with the tip, the ramp, and then the dual blades. I think most of you have seen the procedure, whether you've done it or you've seen it in video, but I am going to show some of the particulars with treatment so you get an idea of how we approach it. Prior to going into the operating room, the University of Colorado partnered with New World Medical, and I think many of you know New World Medical. They're the manufacturer of the Ahmed valve as well as ClearPath and several other devices. You can actually see, just out of interest, that's Dr. Ahmed there in the corner, standing in the office in California together. So he was an early mentor, and we approached the company and said, hey, we'd love to partner. And we started doing these basic studies, looking at the histology of differentiating the dual blade versus an MVR blade and versus other devices like trabectome. And what we found is our hypothesis of creating dual incisions would result in a cleaner removal of trabecular meshwork. You can see MVR blade on the left-hand side. It has an incision in the trabecular meshwork, but it also, unfortunately, is very hard to gauge the depth of the blade cut. So you also see an incision in the scleral tissue, which you'd like to avoid because of the inducement of inflammation and then the potential to reanastomose, reapproximate the trabecular meshwork. Whereas on the dual blade side, you can see from scleral spur to the tethering of the TM remnant that it's quite clear, not a lot of leaflet left behind, and it would be very difficult for the trabecular meshwork to reform in this area as opposed to the leaflets here reconnecting. So it was quite exciting to get these early findings, and we were able to push this through the FDA and into clinical practice, into commercialization. Here's an example of the procedure. This is John Berdal actually doing his first case of KDB. I always like to show the first cases. Very good at gonioscopy already because he's a good MIG surgeon, and you see what's called the outside-in maneuver, going from left to right, and then reversing and going right to left. And as he meets in the middle, you can see the strip of trabecular meshwork actually like Velcro on top of the iris. Sometimes the trabecular meshwork will come out with the device, which you'll see in the next video. In this case, you just go in with your capsulorhexis forceps, you grab the trabecular meshwork strip, and it comes out in whole. You see the reflux, which is a good sign. You want to see that you've actually unroofed the canal. Here's an example of Paul Singh doing the procedure. Also, I believe his first case. He does a longer swipe from right to left, and then a shorter swipe from left to right. And you can see in this case, the trabecular meshwork is actually adherent to the blade, and it comes out with removal of KDB. And you see the nice wall, anterior wall of Schlemm's canal there. So let's talk a little bit about clinical data. Glaucoma specialists are notorious for loving data. We want people to prove to us that things work. New World has been very good about publications, not just for KDB, but for all of their devices. KDB was launched in the US at the end of 2015. So we're in year 10 of KDB now. And there are over 110 publications with data, including prospective data, RCT data on KDB. This is one example early on, just looking at excisional goniotomy with KDB prospectively for 12 months. I like to show this study just to show that no matter what the starting pressure was, whether it's around 20.5 or 13.1, these patients at 1 year all end up around the same place. Typically in my hands, it's somewhere between a pressure of 12 and 14. That's typically where the patients end up. You remove that blockage to the egress of aqueous humor, and essentially most of these patients come to that level of IOP no matter where they started. I also like to point out that it's not just primary open-angle glaucoma. This also works in patients who have chronic angle closure. This is a study by somebody who many of you know, Cyril Dureraj, who's a close friend. He works at Mayo Clinic in Florida here. And he did a study combined with some colleagues in Vietnam looking at the results in patients who have chronic angle. And you can see quite a dramatic decrease in the pressure. In my practice, this is actually standard of care. When we're going in to either do phaco plus or in a standalone patient with chronic angle closure, we'll do goniosyncytialysis combined with KDB goniotomy. And these, our results are very similar to what Cyril got. We have a very active uveitic glaucoma practice here. My partner, Mina Pancheva, has a special day just for uveitic glaucoma. It sounds like a very fun clinic, I'm sure, to you. And she's utilized KDB in these patients and has also seen a dramatic decrease in pressure in those patients once their inflammation is stable to go in and do a goniotomy. In these patients, they respond very well to excisional goniotomy. Now, from a study standpoint, we always look at RCTs. We always like to look at comparators. So a few years ago, we started a study looking at KDB phaco versus iStent phaco. One of the big debates in MIGS today is, should you put an implant or should you try and do something that's implant-free? My practice has been focused on not leaving anything behind. So not doing an iStent, not doing a hydrus. If they work in your hands, of course, continue doing it. I'm not saying that you shouldn't be doing it, but my preference is to not leave anything behind. So we did a prospective study here looking with 1 year of follow-up on patients who were randomized 1-to-1 to either phaco KDB, phaco iStent. And what we found is that significantly more KDB eyes, KDB phaco eyes, 93.7%, compared to the iStent phaco eyes at 83.3%, met the primary outcome of reduction greater than or equal to 20% of IOP lowering, or the IOP medication reduction of greater than or equal to 1 at month 12. And you can see that these patients had a range of pressures. So the average baseline pressure was around 18.5. So we're not talking about patients like the iStent or Hydra studies that started off in a mid-20 pressure. These patients started off at a lower pressure. But yet, both of these devices, whether it's KDB excisional goniotomy or iStent, lowered pressure quite effectively. They both lowered dependence on medication quite effectively, with a slight advantage to KDB. And of course, the advantage from my standpoint, with my biases, I think very well known, is that you got this lowering without leaving something behind in the angle. We still have a lot to learn. The interesting thing about KDB is that it started in the laboratory and made its way into the operating room. And now we're taking what we're learning in the operating room and taking it back to the laboratory, which is something that has been a lot of fun. So we're harvesting these strips of trabecular meshwork, and we're working with other companies who are studying trabecular meshwork, and we're also doing our own internal studies to do histology and really understand the trabecular meshwork of these patients. Historically, it's been very difficult to get harvested tissue from eye banks that are properly phenotyped, that we understand exactly what the history is. In this case, we can do that with our patients and get fresher samples, and it's been leading to multiple studies. One of the things that we noticed early on is that other surgeons started sending me strips of trabecular meshwork, and we started collecting some of these. And in this case, this is a case example of another person that I think a lot of you know, Devinder Grover, excellent surgeon, works with Ron Fellman. They pioneered GAT procedure, and they did a lot of GAT. And many of those, of course, like all glaucoma procedures, tend to fail. So when the GAT procedures failed, they went in and did KDB, and they would harvest some of the trabecular meshwork. And this is a sample that they sent me. And you can see from the histology, what we noticed was that the material around the tissue that's collateral, proximate to the trabecular meshwork was, it looked like there was desmetization, kind of like a basement membrane that was overlying the trabecular meshwork, and also a loss of trabecular meshwork cell and fibrosis, especially around the area of prior procedures. So we looked at this and we thought, I wonder if this is happening with other things. This is a case, this is me doing the surgery, an old eye stent that I put in that looked like it was displaced posteriorly. So I went in and I took the eye stent away and I started doing a KDB to further lower the pressure because the pressure had gone back up after a year or so from the eye stent implantation. This felt very different to me. I actually felt resistance going through the trabecular meshwork, whereas typically I don't feel much resistance when I'm removing the trabecular meshwork. And you can see me here removing that strip, and I can see that there's desmetization that's attaching to the trabecular meshwork strip. And I'll zoom in on it here so you see it more clearly. It was vascularized with desmetization that was around the area of where the eye stent was, exactly in that area, and then to the right of that area. So we harvested the tissue, and then we started harvesting tissue from every patient where we removed an eye stent to do KDB. And we found very consistently that there was a fibrotic tissue leaflet over the device and around the device. We published this in Journal of Glaucoma a few years ago, showing that several cases had exactly the same view. And what I think is happening here is pretty clear. Just like anything that you put around the eye, whether it's an Ahmed valve, a clear path, anything that you're putting in tissues around the eye tends to want to envelop it. It tends to want to cover it, and that's exactly what's happening here. So we have a lot more tissue samples with other devices that we're looking at, and it's a pretty exciting thing to go back to the laboratory where KDB started. I think one thing that I can say pretty clearly is that the dogma that we previously believed in, that goniotomy doesn't work or it might be inappropriate for adults, has been completely disproven. We have over a decade of data with excisional goniotomy with KDB showing that it's quite effective and lasting for several years. We go out to 5 years now with some of our publications showing the efficacy. Now let's get back to the story of where we are. I'm pretty happy that we can tailor the therapy. I will say that in the United States, and I'm told that it's very similar around the world, that despite these new approaches, only a minority of cataract surgeons are engaging in MIGS procedures and angle procedures. So most of our advances have been surgical. And that might be interesting to a lot of us who've been in practice, because when we look at our therapeutics, like our prostaglandin analogs, for example, they're quite effective. Most of the problem is the external sort of side effects that happen with hyperemia, but also the patients are not taking their drops. So what can we do outside of MIGS to get many more surgeons in the sandbox of treating glaucoma at the time of cataract surgery? And that's the part that I'm going to go over here over the next few minutes. I'm going to go over this middle section quite rapidly just for the sake of time, but there have been other devices that looked at guided administration of pharmaceuticals that are patient-independent, physician-administered, device-guided therapies. This is my wish list. I'll just in short say that if it's not an implantable, if it's an insert like a punctal plug, I'd like it to last for at least 3 months. If it is something that's invasive, I'd like for it to last at least 2 years, because you're not going to go back in every 2 years. Everything else I think is quite intuitive here, but that's kind of my bar for what my wish list is. These are the different gap therapies that, partial listing, not complete listing, that have come either in studies or made it to the market. I'm just going to touch on each one of these. So one is punctal plugs, and punctal plugs have been explored for treating glaucoma for quite some time. So many of you might know the name Jean Dewan, retina specialist, previously at Hopkins, moved around a little bit to a couple of other places, is now in San Francisco. He developed one of the first punctal plugs for drug delivery more than 25 years ago, I believe. So this is something that isn't a new field, and other companies have tried to do this. Unfortunately, the retention of these plugs has not been good. Many of these plugs fall out. It also cannot carry a big payload of drug. It doesn't tend to last for more than just a handful of weeks. Subconjunctival injections have also been tried. Anacortev acetate was a drug that we studied here in the US. I was part of the trial that Alcon did. It was injecting the medication subconjunctivally. And that picture that you're seeing in the upper right-hand corner is the milky medication 360 degrees after injection. Cosmetically not very appealing to the patient. Also, when you go in with a needle to inject and then you remove the needle, a lot of the drug was actually coming out of the needle tract, so it was very hard to kind of program in the dose. Of course, if you inject over and over again in this chronic disease and the patient eventually needs a trabeculectomy, now you've made the success rate of your trabeculectomy. The one that did make it to the market is this one. This is Durysta, initially called BIM-SR, sustained release, now called Durysta on the market. From what I understand, it's not available in the Indian market. It's been available here for several years. Now we're talking about something that is invasive. So you're injecting it into the eye, into the anterior chamber. The unfortunate thing here is in the studies, even though it's found to be quite effective for several months, there was also a high rate of corneal endothelial cell damage from the pellet hitting the back of the cornea. And for that reason, from an approval standpoint, the label says that it should only be used one time per eye, which isn't ideal for a chronic disease. It does have a niche area, maybe patients where you're trying to give them a drug holiday. If you're trying to buy some time before surgery, those are areas where it has some utility. I will say that a significant number of patients after one or two of these will actually have lasting effect because of the tissue remodeling. So it's an interesting point to keep an eye on. And I know that Allergan or AbbVie now is working on potentially changing the label so that you can get repeat applications, which I think would be a good thing if it was found to be safe. This is the new kid on the block. This is iDose from Glaukos. We've been waiting to see this for quite some time. This is a titanium barrel with a harpoon feature on the end that gets implanted similar to a MIGS device, so it requires angle surgery skills. Once implanted, it delivers travoprost in this case for several months. The phase 3 trials have been published. It was found to be non-inferior to timolol at 3 months. It looks like towards the end of the first year of follow-up that it loses that non-inferiority to timolol. So we'll have to keep an eye on this and see what further data show. I think this is a good idea to deliver drug. It does, like I said, require gonioscopy skills and the ability to implant across the trabecular meshwork through the canal and into the sclera. And we'll have to see what's required for readministration. Similar to Durysta, at this point, iDose does not have a label for removal and reinsertion. So we'll have to see if they're able to expand that label. Now, for my wish list, we don't quite hit all of the wish list that I had. Safety and efficacy profile equal to or better than PGA. We're not really getting the efficacy to be better than a PGA. And then a lot of the therapies that we have today don't last as long as my wish list. The punctal plugs fall out, so they're not lasting 3 months. The intraocular inserts or implants are definitely not lasting to 24 months. So I'm hoping that we can do better. So let's talk just very quickly about the patient part of this. We don't talk much about how patients respond. I was very curious to see if patients would be accepting of a different therapeutic if they were using drops. And I was surprised to see that almost half the patients would accept something that was more invasive if it helped them decrease or avoid the use of topical medications. Patients in general don't like taking topical therapies. And a second study that we did with Pradeep Ramalu at Hopkins was to ask all the patients in our clinics if they would accept specific types of alternative therapies. Most of the patients said that they would accept something other than a drop if it obviated the need for added surgery. So very similar to MIGS, if you could go in and do it at the time of cataract surgery and you're in there anyway, patients were very accepting of that. Or if it demonstrated greater efficacy than the drops that they were using. So we know that patients are looking for an alternative. In addition to that, this is probably the most striking study that we did when we're looking at maybe studying these devices a little bit more. Patients were asked if they would accept an intravitreal injection for glaucoma over their drops, and 46% said they would accept this if it lasted for only 2 months. So if they got a 2-month break from their drops, they would accept an intravitreal injection that would be repeated every 2 months. And that goes up, for 3 months, 62% of the patients said they would rather do that than take their daily drops. So patients, I think, are asking us for something that's an alternative to the therapeutics that they're using today. Okay, so I'm going to talk a little bit about how to really achieve the true promise of the glaucoma therapeutics that we're using. I think we can say very clearly that it's not just about dollars, that these patients have diminished quality of life and we need to meet their expectations. So I'll close off briefly here in the last 10, 15 minutes talking about 2 devices that we're working on here at the university. The first one is the CU lacrimal system. This is a device that lives in the punctal anatomy through the canaliculus and into the nasolacrimal sac. And I think we should revisit this area as a potential landing spot for some of the therapeutics that we use. We know that the punctum is a really good spot for insertion of punctal plugs or devices that can deliver drugs. And it's very accessible at the slit lamp. You don't have to go to a procedure room. You don't have to go to the operating room. It's quite safe. Everybody knows how to do it. We learn how to do it as first-year residents in training. Optometrists do it routinely. So it's something that's just kind of waiting for the right system to marry with it. What we've done is we've tried to approach it from a very different angle. We approach the punctal anatomy very similar to a punctal plug. We're doing it at the slit lamp, but we've taken the punctal plug and we've stretched the distal end so that the terminal end resides in the nasolacrimal sac. At the terminal end where the nasolacrimal sac is, the device is actually a balloon. Once it's inserted, the balloon can be filled with any type of therapeutic. In the first case of what we're studying, it's a glaucoma therapeutic, a prostaglandin analog. At the proximal end where the tear film is, the faceplate is plugged by a hydrogel. And that hydrogel limits the egress, the exit of the fluid from the balloon through the device and onto the tear film. That hydrogel can be tuned to the specific therapeutic. So if I want to deliver 1 microliter per day or 6 microliters per day, I can tune the hydrogel to go faster or slower. And now we've solved two of the major issues with traditional punctal plugs. We now have 100% retention because the faceplate is holding at the proximal side, the balloon is holding at the distal side. There's really no place for that device to go. And we have more consistent and long-term delivery because we can put a lot more drug in the balloon compared to what you can put in a punctal plug. This is just an animation to show you what it looks like. The device, this is again something that we can do at the slit lamp, goes in through the punctum, through the canaliculus and common canaliculus, and then into the nasolacrimal sac, where the catheter is then used to inflate the balloon with, again, any drug. And once the catheter is removed, now the hydrogel is actually limiting the flow, and it delivers for glaucoma for up to 6 months. At the end of 6 months, you remove it, you put a new one in at the slit lamp, and you can just kind of keep going. If it's for allergy, before allergy season starts, you can put the anti-allergy medication, remove it at the end of 3-4 months when the allergy season is over. So there's a lot of utility. It's a platform technology that can release different therapeutics. We've done extensive preclinical testing on this to make sure that the balloon functions as predicted. And we've also did initial insertions into patients where we went in and we did bilateral insertions to make sure that it's comfortable, that the patient doesn't feel anything, there's no pain, and that it's retained 100% of the time. And now the effort is to say, okay, what do we do next? How do we get into clinical trials? And right now we've actually partnered with Zeiss for production of these devices. We're in process of trying to scale up manufacturing so that we can take this into clinical trials. And I would say stay tuned for this one. I think you'll be hearing a lot more about this one in the next couple of years. So let's go on to the last technology here in the last 10 minutes or so. So Spyglass Pharma is a company that spun out of the University of Colorado. My partner, Glenn Sussman, and I worked on it in the university and then got funding to spin it out into a company that's now based in California. And let me just tell you a little bit about the company if you haven't heard about it. So we know that 20% of cataracts are done in patients who also have glaucoma. So if you look at the Medicare data in the US, it's very clear that about 20% of the patients have ocular hypertension that can be treated with some type of therapeutic or mild to severe glaucoma. There's very low friction for the patients to accept something to treat their glaucoma at the time of cataract surgery, similar to MIGS. That's kind of the story. We tell the patient you have cataract surgery, we can treat your glaucoma with MIGS at the same time. In this case, what we've done is we've taken a hydrophobic acrylic intraocular lens, and we've placed 2 drug depots on the haptic optic junction. And then from this point, it's very similar to what you would expect from cataract surgery. Traditional off-the-shelf injector through a standard clear corneal incision, post-removal of cataract, putting the lens in the capsular bag, unfolds just like any hydrophobic acrylic intraocular lens, and it delivers drug with a payload of 3 years of bimatoprost. So This is the animation, and I'll show you a surgical case here in a little bit, but it gives you an idea of sort of what the idea was when we first started. We went into the first in human study in Central America, and we did low, medium, high dose, not trying to go far away from what Durista was delivering. So we had an idea of what we were targeting. And publicly, we have announced 18-month data. We'll announce further data coming up here in the next couple of months. But at 18 months, we see that almost 44% IOP lowering. All the patients are off their medications, and the visual acuity is exactly what you would expect in a study like this from a leading commercial IOL. We're very excited about this. The doses didn't show much dose separation. We didn't think it would because, again, we stayed very close to what Durista was delivering. This allowed us to move forward with further testing. Here's a surgical case. I just want to show you the loading of this lens, and it's backloaded in a cartridge similar to what you would with any backloaded intraocular lens. You can see the pads there on the haptic optic junction. The trailing haptic is then folded over the optic as it usually is, and then pushed into place. This is then taken and attached to the plunger side of the injector. And the next thing you'll see here is post-removal of cataract going through a standard clear corneal incision and then injecting the lens into the capsular bag. When you see that part, what we've asked the surgeon to do is also move the lens around. One of the early questions that we got was, would you be able to take the lens once it's in the bag and move it around if it's a toric lens, for example, without any limitation from these pads? And that's what you'll see here. So going through a standard incision, injecting into the capsular bag, you'll see the leading drug pad open up, and then there's the trailing drug pad. And the lens unfolds. The flat time of this lens is exactly the same as commercial IOL. So you don't see any springing. You don't see any difficulty with positioning the lens in the capsular bag, making sure that the pads are close to the equator. Of course, once the pupil comes down postoperatively, surgeon can't see the pads. The patient doesn't notice anything from a visual standpoint. Clearing the viscoelastic from behind the lens and in front of the lens looks like standard cataract surgery from this point forward, right? So the minor differences are related to the pad getting underneath the anterior capsule and positioning it in place. But pretty standard cataract surgery. Any cataract surgeon can do this with available equipment. Platform technology is something that's maybe overused in innovation, but I think this is one good case for it, that we can do different medications, whether it's for glaucoma, we can do dropless cataract surgery with NSAID and steroid. We have a therapeutic for macular degeneration that delivers over a long period of time. And all of these are currently being studied at various stages in development. This is the team. I think it's always just good to mention that this is a very big team. So Spyglass has over 40 members in the company today. Everybody has worked very hard to get us to where we are today. We just announced in the past couple of weeks that we finished enrollment of our phase 2 trial in the US. So the first in human data I showed, now we've done a phase 2 study in the United States. And this year we're about to start with our phase 3 trial. That'll be a global study. Should start sometime this year. We have completed our Series C raise. Again, we completed the enrollment of the phase 2. And from a phase 3 standpoint, I think if you look maybe towards the second half of this year, that's when you'll start to see more activity in this space. So a lot of us are talking about AI these days. We're publishing a lot on ChatGPT and how to use different AI systems in our clinic. I put the transcript of my talk on drug delivery into ChatGPT, and I asked it to give me a summary. And I just want to point out one thing that I think is extremely important that I never said during the talk, but AI kind of figured out that maybe I should have. And that is the fact that it's not just the clinician, it's not just the researchers. We need multiple people to get together to really push this forward to clinical use. Clinicians, researchers, regulatory experts are key to help bring this across the finish line, and not just in the US, but regulatory barriers are quite high in countries around the world. Industry members are needed for guidance as well as funding. And then venture capital, it takes a lot of money to get to drug delivery. I can tell you a Series C raise for Spyglass was 90 million. So it takes a significant amount of money to get across the finish line. And one thing that I can say today that's very exciting, similar to how I felt about MIGS a few years ago, for drug delivery, I think now the promise is reality. Thank you very much, and happy to take any questions.
Thank you so much. I can never stop getting amazed by any interaction with you, either individually or on a larger platform.
I
The
appreciate that.
summary, the summary by ChatGPT was amazing, as generic as it could be, but actually speaking the truth about the promises becoming reality.
Right. Yeah, it's pretty
And
exciting.
I'll be very happy to take questions from the panelists, starting with Dr. Satyan.
Yeah, thank you so much, Professor. We have three panelists today: Siddharth, Dr. Devendra, and Dr. Shika Gupta from RPC. Dr. Devendra from Arvind Eye Care System, and Dr. Siddharth from LV Prasad. Siddharth, I think you can just start having the discussions. We can.
Great.
Yeah.
I have a lot of questions, but one main thing is why does bimetroprost figure as the first drug of choice for a lot of these implants?
Yeah, I think there are several reasons for that. We have a wealth of data on bimatoprost, both as a topical therapy as well as an intraocular delivery platform. So that gives a lot of speed to the initial parts of the testing. We can run much faster. That's one. Two, bimatoprost is quite potent. We know that it works really well, but we can get a lot of the drug into a small space to deliver over a long period of time and only have to deliver a very small amount to get efficacy. So that gives us a long runway. We can go much more than 3 years. The first product is going to be 3 years, but we can take this out for several years. It's also quite stable in the eye. There are some drugs that you can put into the eye that aren't very stable, but bimatoprost in particular is one of the most stable drugs that we can put in the eye, and it stays very stable in that environment of 35C. And we know this from other companies as well as our own internal testing. So there are a lot of reasons for it.
Okay. Is that after 3 years' time you reload the drug?
So once that's probably the number one question that investors were asking when we were going through the development. The initial generation 1 device that goes into the capsular bag, we never want to touch, right? That's functioning as an intraocular lens. The pads don't shrink, they don't change mechanically. So the lens position never changes, and we can fix vision, which is the target there, of course, when you're doing cataract surgery. We do have another device that allows for treatment after the 3 years are over, and then that device can be exchanged every 3 years or so. We're just not sharing what that product is publicly yet, so you'll have to invite me back when that one's out, and I'll give a talk on that one.
Every month we can invite you, not a problem.
Yeah.
I have a question. That was an amazing talk and an amazing lifetime of work, Professor Malik.
Thank you.
So I would like to ask, the major questions are on drug delivery because that's the upcoming topic, and we've been doing KDBs. For about the drug delivery, I wanted to ask. So you have done punctal, you have a personal experience with punctal plug kind of delivery and iDose kind of delivery, where intracameral or trabecular meshwork delivery is done. So do you think there's a difference in the amount of side effects or adverse effects, that ocular allergies, and what are the different side effects that you see with intraocular delivery, which is not seen with the extraocular kind of like punctal plugs delivery kind of system?
Yeah, I got this question yesterday from one of the big investors in the US who had been to a talk where the whole talk was about prostaglandin analogs and how they cause redness. And he was wondering why that doesn't happen with Spyglass. So if you look at Durysta, iDose, and Spyglass, they tend not to have hyperemia to large degree. You don't see the lash growth. You don't see the issues with
That's an added
periorbital fat
advantage.
loss.
Yeah,
Right.
that's
Yeah.
added
So
advantage. Yeah.
I think there are two ways to look at this. I think if you're doing something external, similar to the lacrimal device that we're working on, the big advantage there is we can deliver a very small amount of drug rather than the 50 microliters that we're typically dropping on the eye. In addition to that, we don't have to use preservatives. So the benzalkonium chloride that comes with topical therapeutics can cause damage over time as it is soaked up by the tissue. So it's still going to cause some of the issues that we see with topical therapy. You're still going to see some hyperemia. You're going to see the lash growth. You might see, some of the periorbital changes. But it should be less when you're delivering less drug, and you shouldn't see the skin issues that you're seeing when the drops go onto the skin with hyperpigmentation. When you're in the eye, a lot of that is actually taken off of the table immediately. We don't see any of those things. So when we're talking to the patients, when eventually these things are commercialized or with iDose today, I think that's part of the conversation for patients who are having drop intolerance, and that's the major reason for their non-adherence.
Mhm.
So it opens up a whole new world to how we use these very effective therapeutics without being limited by their side effects when they're topical.
Yeah, that's an added advantage, I would say. So another thing, we have systems now. You have systems of drug delivery where we are able to sustain the drug for 3 months, 6 months, and now 3 years with the IUL-eluting drugs. So is it the change in the dose only, or some pharmacokinetics because of the material of the implant that you are just for the sake of our knowledge? So how has this been possible?
Yeah, so it's a very basic answer. It's being able to put more of the drug when you're in the right space. So when you're in the capsular bag, you have a lot more space than if you're in the angle, right? The angle is about a millimeter or so. If the iDose is bigger than what it is today, it's going to be much more likely to hit the cornea or the iris. And that, of course, raises a bunch of concerns. In the capsular bag, we have a lot more space to even make it bigger than what we have today and still be completely fine through a small injector and a small incision. And with that, if you use the right diffusion membrane, then you can really regulate the dose and have that depot deliver over a long period of time. That was so the first 2 years of Spyglass, that's what we were working on to make sure that our drug-eluting pads could do so almost at zero order kinetics. So we're delivering, and our surprise actually was that we had to decide from a commercial standpoint, do we want to deliver for more than 3 years, less than 3 years? The limitation was really about use rather than the device itself. I'll
Yeah.
tell you just very quickly, when we went to doctors and we said, what do you want? The doctor said, if you just give me 12 months of delivery, I'll implant this in everybody. And we thought, no, that's not enough, right? Because you don't want to have to do something every year. When we said 2 years, they kind of looked at us like, can you really do that? And oh, that would be great. Definitely use it in everybody. When we got to 3 years, they said that's enough. You don't have to you don't have to give us more than that. It also, from a regulatory standpoint, to get this approved, when you go much more beyond 3 years, different questions start
Yeah.
coming up. So it was a really good spot for us to land in.
And does the drug concentration, it decreases over time or it remains stable? It is zero order kinetics, so it remains stable over 3 years?
So far, I can tell you both from our animal testing, our benchtop testing, and from what we're seeing in efficacy, and we have further data, much more than the 18 months that I showed, it is behaving in a very stable way. In the laboratory, very flat, not decreasing. We're out greater than 3 years now.
Okay.
Animal studies showed correlation with that. And then from an efficacy standpoint, we're seeing that the patients remain controlled for an extended period of time off of their topical therapeutics. Right? That's
Wow.
a key
Great
thing.
question and a great answer. And Dr. Devendra has been waiting for a long time.
No,
And yeah, please.
yeah, Dr. Malik is a very informative and excellent talk. I'm very fortunate to
Thank you.
part of it. So, there are several devices came and they tested. Only my concern is this: what about the cost? How we can cut the cost of the such devices? Because ultimately, in the maybe US, some part of the population can afford it. So, how you can reduce the cost of the such drug delivery system?
Yeah. And that's a question that comes up with the MIGS space as well, right? So it's a very important discussion to have. When iDose came out, I was sitting here actually in my office, and I saw the sticker price come out, and it was in the US, iDose is almost $14,000, and I was very surprised by that. And if you look at it from a health economic standpoint, you're not using your drops, you're well controlled, the patient doesn't have the adverse events, maybe their disease is progressing more slowly. That starts to make sense in the US market, that cost, because you're taking other costs off the table. Globally, the companies are going to have to figure it out. You can't take this into some markets and charge 14,000. It's just not going to get used. The healthcare system can't afford it. So this is where other alternative therapeutics come in. And it was the same story between KDB and iStent, for example. So KDB is much more affordable than iStent. And we were able to get KDB into different markets around the world much more rapidly because of that. So they were much more accepting of it. Different markets hold different prices. So at some point, you have to get to a place where the patient's accepting, the doctor's accepting, the healthcare system can accept that, and that's going to be very different in each country. It's a very important part of the conversation.
Right. So my second question is regarding the KDB. So I'm shifting the gear now, coming to the what we can afford. So my second question to you, you talk about the outcome of KDB in angle-closure glaucoma. So what's your experience in the chronic angle-closure glaucoma, and how long this effect will sustain post-KDB?
Probably one of the bigger surprises with KDB is the efficacy in chronic angle closure. It wasn't something that we set out to study early on, but I started getting reports from friends in Vietnam who were using it extensively for chronic angle closure, combined with goniosyncytialysis. In my hands, if you just do goniosyncytialysis, you get pressure lowering, but it's not as effective across the board. Some patients see no change. Some patients have an elevation in their pressure. Some patients have a significant decrease. What we've noticed in combining KDB with goniosyncytialysis is that almost all of the patients have a significant decrease in their pressure because most of their trabecular meshwork is no longer functioning because of the adherence that was in place for, in many cases, multiple years. So what we're seeing is a significant decrease to the low teens, and that has been sustained uniformly in greater than 80% of the patients for multiple years. We have patients that we followed now in my practice up to 10 years because KDB has been out for 10 years. And we're seeing, of course, with every form of glaucoma surgery, you see some failures. But with chronic angle closure, it's been quite effective. And I think it should be standard of care in these patients to do goniosyncytialysis with removal of the trabecular meshwork.
Thank you. Thank you. Nice to hear, because I do a lot of KDB in angle closure glaucoma. Only
That's
problem,
perfect. Yeah.
everybody I do,
Same.
questions for me. How long it will sustain? I think I got the answer. So my second question related to it, what if KDB fails, what next, in your opinion?
I think that's one of the things. We're in the golden age of glaucoma surgery now. So we have multiple things that we can do. In the angle, typically when I do KDB, I'll do about 3 to 4 clock hours. So you can go in, we have access to ECP, endocyclophotocoagulation. I do quite a bit of ECP. You can do more of a goniotomy, which tends to be effective. You can go in and do canaloplasty with a trabeculotomy in the remaining canal with trabecular meshwork. Or you could move on to some of the more involved, not really MIGS, but sort of MIGS like Zen or PreserFlow, which we don't have in the US quite yet, but we have Zen. What I'd like to do, my whole career has been to avoid a bleb. I don't like blebs. I don't want blebs. But in some cases where MIGS has failed, then you can go to something like a Zen, or in the US, we now have ClearPath, and we have a smaller tube ClearPath. There are a lot of options that we have right now to go to. Whereas before we would go straight to CPC because we did everything so quickly. Now we have options.
So is related to what is your experience? If KDB fails, can GAT work? Your experience or your opinion?
Can GAT work? Yes, GAT can work. So in my hands, I typically go in and I'll do an incision that is supranasal or superior, and I'll do the remaining angle with a KDB. But you can also quite easily go in with a suture and do a GAT procedure and do the temporal trabecular meshwork. I don't do a lot of GAT. Part of it is that there's so many other options to do that are maybe less invasive than that. But we do, in our practice, quite a bit of GAT in uveitic glaucoma patients, and that tends to work really well. So yeah, there are options again, and it's what you're comfortable with. If you're comfortable with GAT, then I think it's certainly an option to do.
One more question.
Great. Ted. We'll just get back to you. I'll intervene here and with two of my questions. First, the CU lacrimal based drug delivery system, it's probably aimed at much more than just the glaucoma drugs, right?
That's right. Yeah.
Right.
So yeah, we're looking at allergy medications. Post-operative drops. So there's a drug here that is used for post-cataract surgery with a hydrogel that it's called Dextenza that delivers dexamethasone. So something similar to that would deliver both NSAID and steroid. Also, you can imagine a patient comes in with a corneal ulcer. Now we do antibiotics several drops per day. Patients are waking up. Some of them are admitted to the hospital for therapy. In this case, you can put this device in and it delivers antibiotic 24 hours for a consistent time over weeks as you need. So there are a lot of things you can do with this device.
So the proprietary gel, it's not dependent on the molecule that you want to deliver. It's an inherent property of the gel to elute the drug consistently.
Yeah, the gel is only sitting in that part proximate to the face plate, and the drug, of course, is on the inside. And what you can think of it as a sponge that we can alter the pores in that sponge. I can make the pores bigger if I want to deliver faster, and I can make the pores smaller if I want to deliver it slower. And so
So
then you can tune it to each
every
drug.
drug can be delivered, right? And just one thing, if you have a kind of a dilated balloon and in the nasolacrimal duct, the moment the system gets into the sac, would it interfere with the normal lacrimal drainage system?
So this was one of our initial worries when we went into clinic to make sure. So it was designed in such a way that tears will still flow past from the superior punctum past the common canaliculus and into the nasolacrimal sac. I'm sure some patients, when this is more widely used, will have epiphora and maybe will not be able to use this. I think that'll be a small number of patients. The majority of patients and everybody that we've had this in so far did not have epiphora because of the specific design to allow for tear flow.
Great, great. I'm very excited for this. Any burning questions from Shikha or Devendra?
Siddharth, I have one question to Professor.
Right.
We all been in India, typically we all reuse your blade. So why does this blade goes off? I mean, it becomes blunt in a few cases. Though the question is based on it is a metal, and when you just strip down few cases, why does it go a little blunt? Is that any specific things?
Why does it go blunt? Is that?
Yeah, after two or three cases.
So there are a couple of really important things here about reuse. And there are some things in ophthalmology that we reuse and maybe can survive with some of the tubing, maybe in phaco. With KDB, you probably don't want to reuse it because of two things. One, the blades actually are very, very thin and fine. And after you just do an initial treatment, once you investigate the blades, you'll see that it's not something that you can use 2, 3, 4 times and still maintain the proper incisions that you're aiming for. It becomes more like a blunt tool. So you're taking away the advantage of the dual blades. The other part that is probably more important is that there's a pocket between the two blades. And this pocket retains a significant amount of the trabecular meshwork and trabecular meshwork cells. We know this from our studies when we do a treatment and we take the blade and we wash it vigorously and we put it under our advanced imaging devices, we still see tissue on the inside and we can still see trabecular meshwork. So if you try and wash it and then reuse it, you're introducing foreign cell material into eyes. And unfortunately, this did happen in one practice outside of the US where the eye developed severe inflammation. And I got a call from the surgeon and I was talking to him and he said this was the third time he was using it, that he felt like he washed it really well. And we got the device and looked at it, and sure enough, there was still cells on the inside. So I would ask you not to reuse. I know there's a big debate about reuse, but in this case, I think it's a patient safety issue.
Okay.
Great. Dr.
Thank you.
Satyen, time for the closing remarks in interest of time.
Last question. Siddharth, last question.
I want to answer Devendra's last question. So
Yeah.
Go ahead.
So there's a lot of buzz about the 180-degree versus 360-degree. So right now, what do you think about the treatment dose response? How do you plan out only 4 clock hour? If we going to do the 180-degree, did we get the more IP reduction? What do you think? What are your thoughts?
So I'll keep this brief because we do have extensive knowledge now and published research on multiple devices, including on the some of the canaloplasty treatments that 180 gives you of a trabeculotomy or a goniotomy gives you as much efficacy as 360. I can tell you in our studies, both in the laboratory in perfused cadaveric eyes as well as in the operating room, once you get beyond 4 clock hours, you do not get increased efficacy. And for that reason, our standard practice here is to go 3 to 4 clock hours and no more. I think that the published literature actually kind of conclusively answered that question, that more does not mean better in this case. I think you can stop at 4 clock hours.
Can I
Thank
add
you.
to this? Can I add some sentence to this?
Vika, I think we can keep on going on with these
Okay.
questions definitely. Just to respect our audience, we would like to close this session. Our sincere thanks on behalf of Glaucoma Society of India to Professor for taking all his time off and giving this wonderful lecture with a lot of clarity for all of us. So once again, we would like to thank you so much for sparing your time for us and giving this wonderful lecture for us. And we all expect you to be here in India for our annual conference in September 12th to 14th in Bhopal. So I'll
I'd
send
love
you
to visit with you guys.
for you to accept that. Thank you so much once again. And thank you all the panelists for being here with us and then asking wonderful questions for our audience. Thank you so much. Good night.
Thank you all. Yeah,
Thank you.
good night.
Good night.